Wait, so how does coronavirus work? Hi, my name is Batul Saruri, and today I'm going to be talking to you about coronavirus and how the COVID-19 virus is able to infect and kill our healthy cells. Before anything else, it's important to understand that coronavirus is just a general name for a group of viruses. There have been other coronaviruses that have occurred in the past. This includes the Middle Eastern Respiratory Syndrome, or MERS. This occurred in 2012. In 1912, there was a very interesting case. German veterinarians realized that a lot of the farm animals were getting sicknesses. For example, lots of the chickens were getting bronchitis. This would prove to be extremely useful in later years. The first human coronavirus was identified in the 1960s by the research of Dr. David Tyrrell and the virus imaging pioneer by the name of June Almeida. From a single sample of nasal washings from a young schoolboy in Surrey, Dr. Tyrrell realized that the disease was probably caused by a new virus. Because of this, he decided to send a sample of the virus to the best electron microscopist in Britain at the time, by the name of June Almeida. When she examined this virus under a transmission electron microscope, she realized that it had a very distinctive crown, or corona, which she realized was very similar to the structure of the virus that was causing bronchitis in the chickens. So, Tyrrell and Almeida called this virus the coronavirus, which is what we now call this group of viruses that includes MERS and COVID-19. We can now start to understand more about the virus and how it is so adapted for its function. Firstly, it has lots of spike proteins on its exterior. These spike proteins have binding sites that can bind to a specific receptor on a human cell. The specific receptor is called the ACE2 receptor. What is extremely concerning about the COVID-19 virus is that the shape of its binding site is extremely complementary to the ACE2 receptor. This is extremely significant because it means that there is a much higher likelihood of a successful collision occurring between the receptor and the binding site on the spike proteins. After this, the next step is virus-host membrane fusion. For this to occur, the spike proteins on the virus must first be processed so that they can bind to the protease TMPRSS2. On the spike proteins of the virus, there is a boundary called the S1-S2 boundary. It is thought that this boundary cleaves or splits by the use of an enzyme called furin. This is an enzyme that is possessed by many of the cells in our body. The enzyme furin can help us understand why coronaviruses often attack the respiratory tract. It's because there is a very, very high abundance of this enzyme here. Once this fusion has occurred, the virus is then able to inject its RNA into the host cell. This RNA is then able to replicate and be translated into proteins by hijacking the metabolic machinery of the host cell. This genetic information and protein can then be assembled into new viral particles. Eventually, the cell gets so full of these viral particles that it bursts in a process known as cell lysis. This shows us how the COVID-19 virus is able to infect and kill our cells. I hope this video helped you to understand more about coronavirus, and specifically how the COVID-19 virus is able to cause so much damage to our cells. Bye!